Sprouting of bladder afferents following spinal cord injury is independent from the lumbosacral cord highly repulsive environment. Have no affiliations or conflict of interest to disclose. Neurogenic detrusor overactivity, or NDO, emergence after a spinal cord injury has been linked to neuroplastic changes in the lumbosacral cord neuronal pathways, particularly sprouting of bladder afferents. Mechanisms regulating this abnormal sprouting might involve changes in the expression of inhibitory cues regulating axonal growth, such as Nogo-A and Phosphacan. However, it is presently unclear whether bladder afferents recognize these repulsive cues and respond to them by regulating the expression of their receptor complexes, such as Nogo-receptor 1 and P75, Nogo-receptor 1 and 3, and PTP sigma-LAR receptors, thus adjusting growth responses. Adult female rats were used in this experiment, and animals were divided into a control group in which the spinal cord was left spinal intact, and two other groups underwent largely incomplete spinal cord transection at T8 T9 spinal segments and were left to recover for 7 or 28 days. At each time point, lumbosacral spinal cord segments, L5 to S1, and associated DRG neurons were collected. Lumbosacral cords were processed by Western blotting to evaluate the expression of Nogo-A, Phosphacan, and GAP43, which is a marker of axonal sprouting. Total RNA from DRG neurons was extracted, reverse transcribed, and analyzed by qPCR to evaluate the expression of Nogo-A receptors, which is the complex Nogo-receptor 1 and P75, and Phosphacan receptor complexes, Nogo-receptor 1 and 3, and PTP sigma-LAR receptors. Concerning the results, a time-dependent increase in GAP43 expression at the lumbosacral cord was found following the injury. Lumbosacral cord expression of Nogo-A and Phosphacan had a threefold increase at 7 days post-injury compared to controls, and returned to baseline values 28 days post-injury. Additionally, in lumbosacral DRG neurons, Phosphacan receptor complexes, Nogo-receptor 1, Nogo-receptor 3, and PTP sigma-LAR receptors demonstrated a time-dependent decrease in expression compared to controls. Nogo-A receptor complex, Nogo-receptor 1 and P75 also showed a time-dependent decrease in expression compared to spinal intact animals. As a conclusion, here we observed alterations in expression of known inhibitors of axonal growth, Nogo-A and Phosphacan at the lumbosacral cord following a thoracic spinal cord injury. Both proteins were overexpressed at the lumbosacral cord 7 days post-injury, even though axonal sprouting continued to occur at this site, which is indicated by GAP43 levels. Despite the presence of a highly repulsive environment, sensory afferents were able to continue to grow, and the expression of receptors was time-dependently downregulated. These results indicate that NDO emergence, which has been linked to aberrant sprouting of sensory afferents, reflects changes in the molecular mechanisms regulating axonal growth.